Hello, my name is Lawrence. We're looking at barium sensing glycerine derivatives for the detection of neutrinoless double beta decay. Neutrinoless double beta decay is a nuclear process that involves the conversion of two neutrons into two protons. The reaction that we usually use to model this is xenon decaying into barium, which is that first reaction in the top left. As it does this, it releases neutrinos and electrons. However, the second reaction is a neutrinoless version of the first one. That one is a lot higher in energy and it's a lot more infrequent. We've never been able to see it experimentally, which is what we're trying to do. We're trying to make molecules that will sense the barium that comes from this reaction. We want to take our molecules and put them sort of on the front face of this time projection chamber that you can see modeled there. An electric field passes through the chamber that will select for the barium ions and push them into our chemosensors. On one of the other faces of the chamber, it will sense electrons as they move through space, through time. We can convert that to energy in order to confirm that we have the reaction that we want. In our previously done computational studies, we've been able to predict that the fluorophores will not exhibit any fluorescence in the absence of barium-2 ions, but they will fluoresce when there is barium ions. The way that it works is you can see the molecule at the top has sort of a cup-shaped binding domain. That is where the barium ions get caught. It's linked via an aromatic linker to the fluorophore, and in our case, we're using either naphthalamide or BODIPI molecules for our fluorophores. The way we're making these is by starting with para-1-fluoronitrobenzene. We reflex that to make our second compound, and then add a protecting group in order to keep the hydroxyls on there from interfering with us adding on a naphthalamide. We take this molecule, we deprotect it, and while we're doing that, we are taking our Calix molecule and putting propyl groups on that. The point of doing so is to keep it from changing its conformations too much. We want to keep them all the same conformation. We take this, we add it onto our previously made molecule in order to put them together. And at the end, we want to replace the bromine at the bottom with a butylamine. This shifts the fluorescence into the range that we want it to be in and sort of allows us to look into maybe putting it onto a surface in the future. The reason we haven't moved too super far in our research is that our glycerine can take on different conformations depending on what substituents are on that lower ring. This makes it a lot harder to control for. This is super hard to purify. We have a whole poster on this that Jackie is going to be presenting. She's kind of co-presenting with me. But as far as for future directions for this research, we want to be able to, being able to detect single ions at a time and have them fluoresce is pretty helpful. We are hoping that we can use our molecules to improve on our previous results. Basically in conclusion, we've identified these molecules as being able to sense the barium ions. We have a pathway to synthesizing them, and most of our aims are to finish the synthesis and be able to use them in future testing. Oh, there's thunder. We want to thank our collaborators. I would like to thank Dr. Foss and everyone else in the lab for their work. Thank you very much.